Buldum hocam.
Doktor Şafi,
Efendim hocam.
bir önceki oturuma katılamadı Züleyha Hanım var. Bu oturumun sonuna onu ekleyeceğiz. Bağlandı burada. Züleyha
Tamamdır.
İdado Muhtar.
Tamam hocam.
Mr. Kroma, how are you?
I am fine. You?
Fine, thanks. Where are you from?
I am from Liberia.
Liberia.
Yes.
Yes. What's your university?
Chukurova University.
Kroma.
Yes.
Biology.
Yes.
Okay. PhD, master,
No, undergraduate.
you're undergraduate. Oh, maşallah. I can see someone, Kausu Sanyang.
Yeah, that's me, Kausu from the Gambia.
From the Gambia. Osman
Yes.
student, huh?
Yes,
Okay. Nice to meet you. Welcome to our Congress.
thank you. Nice to meet you too. Pleasure is mine.
Dr. Shafi, we may start the session. The first participant is here. Yes, how are you?
Osman.
Hocam merhabalar, nasılsınız?
Sağ ol, sen nasılsın?
Sağ olun hocam, nasıl olsun?
And Anissa, where are you from, Anissa?
Hello, sir. I'm from Indonesia.
Indonesia.
Yes, Indonesia.
MashaAllah, nice to meet you. Are you going to present or Dini will present?
I'm to present, sir.
Okay. Okay. I'm Mehmet Çevik, the chair of the Congress. I thank you, all of the participants, for your contribution to our Congress. And this is the last session, session 16 of our Congress two-day program. And Dr. Muhammed Shafi is the session chair. Yes, Dr. Shafi, you may start.
Okay. Assalamu alaikum, everyone. My name is Mohammed Shafi. I'm coming from Comoros Island. I did my PhD in bioengineering at Ege University in Izmir. Now I'm working at Ege Hospital, a private hospital here in Izmir. I'm staying in Izmir also at the same time. First of all, I'd like to take this chance to thank all the organizers of this Congress, especially our Professor Dr. Mehmet Çevik and the teams who are working very hard for every year to organize such kind of program, which is very important in our life, especially in our research domain. For that, as it was mentioned before, our session, it will be the 16th session, which will going to be presented by many scholars. The first one will be our brother Osman Bajinka, and will be followed, but I don't know, Hojam, if Shadman is present. I don't know if he is here. He will be the second one. Hojam, your microphone is off, please.
I think he will present.
Okay. Next to
Maybe
that, our
we can give his turn to
Anissa.
Dr. Züleyha. Maybe she might have problems with internet connection. I think
Okay.
she is connected now.
Oh, I can hear you now. I'm connected.
Okay.
Okay, will be followed by our Dr. Züleyha and followed by Anissa Iktiarani. After that will be presented by our brother Yaizal Urganji, followed by Muhammad Ali, and the last one, Musa Ali Hassan Kroma. So without losing any time, please kindly let's start by our brother Osman Bajinka, please, which will he's going to speak about the respiratory syncytial virus infection disturb the pulmonary microbiome and immune barriers to induce microglia phenotype shift. Please, Osman Bajinka, you can start your presentation. And every presenter will be having 15 minutes, including the question session also. So that means, if possible, you can arrange your time to make it with 10 minutes for the presentation and 5 minutes for question, please. Within 15 minutes, we will stop the presentation and we will go for the next one. Please, Brother Osman, you have the speech, please.
Thank you. Thank you so much, the moderator. And I will quickly share my screen. Yes, is my screen visible? Yes, can you confirm that my screen is visible?
Yes, it's okay, Osman.
Perfect, thank you. Yes, as the moderator rightly said, I will also first and foremost thank the organizing committee and of course our long-serving Professor Mehmet Javik. And I'm also an Turkey alumni. I studied in Ege University for Masters in Microbiology, and I did a graduate coursework in Research Methods and Ethics at İzmir Katikelve University before coming back to my country in 2018. So I met Professor Mehmet Javik, perhaps Professor now, as I call him. So today, my topic will be, of course, I might need to talk a little bit about myself. After my master's in Turkey, I came back to Gambia, where I resumed a lecturer position at the University of Gambia. Then 2019, I traveled to China for PhD. Luckily, I completed my PhD just now. So I'm the newest PhD holder now, and it was a PhD in medical microbiology. And now I'm a lecturer at the University of Gambia. So that's all about me. Today, we're gonna talk about respiratory syncytial virus, which is, yes, this is a virus affecting children or infants and also adults who are immune compromised. Actually, how this respiratory virus disrupts the respiratory microbiome, that is the microbiome in the lung cavity, in the pulmonary cavity. And that changes actually induce a microbial shift, phenotype shift in the brain. So this is, it is a follow-up study or follow-up research or experiment on my PhD thesis, which is lung brain axis. So I will explain a bit about it. What happened is like the leading cause of hospitalization of infants in the developing world is acute viral bronchitis. This is a very serious cases for infants. Approximately up to 80% of the children's lower respiratory tract infections are caused by RNA enveloped virus. So respiratory syncytial virus is one of the type of RNA enveloped viruses, right? So we talking about RSV. Now I will not say respiratory syncytial virus anymore. RSV has been so associated with asthma, myocarditis, and other diseases in the elderly and immunocompromised patients. So it's a very serious disease as well. It may be established that post-RSV lung inflammation, meaning those who are infected with RSV virus, they actually in their lung, there's normally an inflammation. And this inflammation induces neuroinflammation, like what happens in the lung has an effect in the brain. So this is a concept, and this is a prediction that actually has been established. The association of the lung to the brain in the lung-brain axis pathway has associated effects of dysbiotic pulmonary microbiomes and neurological disease. So in this sense, we now understand a lot about gut microbiome, meaning gut-brain axis. You know, a lot of neurotransmitters are produced in the gut, and then the more diverse, the healthier the gut microbiota is believed, and the more diverse, of course, the more less inflammatory cytokines or our inflammatory imprints are observed in the brain. So this is gut-brain axis. But there was no study actually done to consider if there is another biological pathway linking the lung to the brain. So this is what I actually decided to study on, lung-brain axis, to see the pathogenical imprints, the inflammatory effects, the epithelial sheddings happening in the lung cavity, whether they have implications in the central nervous system, that is the brain. So the concept of the paper luckily was published with critical reviews of microbiology. So this is very high impact factor journal of 12 Q1 level. So this is one of the best journals in this category. And of course, we actually at the conceptual form, we actually decided that, or we predicted that the microbiome from the pulmonary cavity, this is the pulmonary cavity, this is the lung microbiota. I will use microbiome sometimes interchangeable with microbiota. Microbiota is the organism or the microorganisms in the pulmonary cavity, lung, or say lung microbiota. Then this microorganism, together with their genetic information, together with their DNA, on everything with their genomes, is called microbiome. So I might use microbiota at the same time microbiome. Please let this not cause any confusion. So this here, if you can see the pathway, this is the lung, and these are the microbiota. So we believe that this pathway actually connects the lung to the brain in different biological pathways. We predicted it could be hormonal pathway, which is both bidirectional. Okay, it could also be immune system pathway, meaning, of course, the microbiota produce metabolites that actually promotes neurotransmitters, or of course the TH helper cells. So it could be immune system. It could also be vagus nerve biological pathway. However, it can also be metabolized neurotransmitters, meaning direct translocation of microbiomes. This from the lung to the brain. These are predicted. And then I made a follow-up study. That's the chapter 1 for my thesis. This chapter tells us that, well, in the lung-brain biological axis, metabolomes, these metabolites, and the pathological changes in the lungs and in the brains of respiratory viruses are similar. They're almost alike. So that shows that, okay, perhaps what happens in the lung cavity has imprints in the brain with regards to respiratory syncytial virus. We found uniform metabolites and uniform pathological changes in the lung and that of the brains of the same mice group, respiratory syncytial virus infected mice. Then we actually found we need to know what are the biological pathways. Is it a direct translocation of RSV or is it a metabolical pathway? This is the chapter 1. Then this pushed me to go to today's presentation, which is chapter 3 of my thesis. There's a need for us to study. Why? Because we hypothesize that RSV-induced dysbiosis in the lung cavity may induce disruption of the immune barriers and induce a shift in microbial phenotype in the brain. Well, this study, present study, aims to identify pulmonary bacterial dysbiosis. So I will explain dysbiosis a bit. In a normal microbiota, the more diverse, the healthier it becomes. The reduced diverse microbial population in the gut or in the pulmonary cavity is called dysbiosis. Dysbiosis means reduced microbiota. So it says that there is a reduced microbiota after RSV infection. This is a viral infection. After the viral infection, then the bacterial population we do see in the pulmonary cavity is reduced. Remember, when you talk about microbiome until now, we are more talking about bacterium because virulum and microbiome are two are, of course, understudied until now. Perhaps in the long run, we'll happen to know what are their metabolites, what are their pathogenicity, and what role do they play in collaboration with the bacterial population. So we believe that, of course, we actually saw that dysbiosis after the RSV infection impact the dysbiosis or impact of dysbiosis on the lung and the brain. So we want to see for the normal group, there is no microbial reduced, but for the RSV group, of course, bacterial population has been reduced. So we want to see what are the effects of this reduced microbiota in the central nervous system. So the study will want to identify lung microbiota mechanism. If there is any mechanism linking to the RSV infection by investigating the differentiation of the immune cells, yes, number 1. And also we want to know whether this actually happens in the brain, meaning in the lung and in the brains of RSV during the infection. Very interesting study. Perhaps we decided to bring some of the materials and methods, animal mice control, animal mice model as usual. There are 3 different groups. One is, of course, 4. One is normal group. Then we have one as ampicillin, which is a normal control as well. Then RSV group, then RSV plus ampicillin group. Ampicillin is antibiotic, right? So we did HE staining to look at the tissue morphology. Immunofluorescence microscope was done to look at the expression of the proteins and, of course, the morphology as well. Then how will I call it? Reverse transcriptive PCR was done. Total RNA extraction was done based on the protocol using the reverse transcription, and then we detect RNA virus. ELISA, enzyme-linked immunoadjuvant assay, was done to detect the antibodies in the blood. Actually did construct 16S RNA library sequence specific to determine species-specific signature, that is the metagenomic. And then I had to use MDA and SOD test kits to detect the oxidative stress. So these two actually will determine whether there is oxidative stress or not. And then the data was analyzed using SPSS 210 and the metabolic analysis t-test for the variance and ANOVA with the LDS-PRS comparison were used for the analysis. The findings are interesting, like I told you. So quickly, I will take you through the images so that we'll be on the same page. If you can see the image A here, this is control, and this is control plus ampicillin. So if you can see the image between control and ampicillin, it's more or less the same, no effect. However, for the RSV group, meaning the ones that are infected with the virus, you can see some epithelial shedding. You can see some kind of inflammation around the end. And this is actually not different from RSV, which one RSV plus the ampicillin group. RSV alone and ampicillin alone are more or less the same. So it tells us that, well, actually, RSV can induce epithelial shedding, but the induction of epithelial shedding is not very different when even ampicillin is added to it. That is for the A. And for the B, with the immunofluorescence microscope images, let's look at the control again, plus control and ampicillin, you could virtually see that there is more or less little differences, meaning it's still the same. But once we take for the immunofluorescence for the RSV group, we realize that, yes, of course, there are differences. There's, of course, the epithelial shedding can be seen, the inflammation can also be seen, and all those imprints are here. And again, this is not also different from the RSV ampicillin. So it is like RSV has the effects of this epithelial shedding, but ampicillin also, even if you add ampicillin or antibiotic, the difference still remains the same. It's not significant. And this is also shown with the RSV titer values, okay, in the long assay using real-time PCR or quantitative PCR. So if you can look at the messenger RNA expression here, the control and the control group and the ampicillin, there is no significance for it. However, you can see that the viral titer has risen with the RSV, meaning the more you infect the virus, the more you infect the mice with the virus, the higher the value will go up. And this is almost the same level. The mRNA titer of mRNA RSV titer for ampicillin, RSV ampicillin is almost equal to this one. The same trend follows for MDA and SOD. So remember, MDA and SOD I bring those ones to determine the stress markers, whether there is going to be too much stress on the mice or not. So you can realize that, yes, with MDA, it is less. Of course, there is a less stress. Again, the control and ampicillin is more or less the same, and the RSV plus RSV ampicillin, there is also no significance between the two. So no significance between the control and no significance between the two groups, meaning, of course, RSV can insert it, and then the ampicillin will not even decrease the MDA level. However, on the SOD level, we realize that the control group, meaning they are free. When no RSV infection, the SOD level is higher, which is a marker in the respiratory marker of stress. The higher, the better for the SOD. And then the lower for the rest RSV and RSV ampicillin means there is a stress induced, right? So I then moved to the long cytokines to see, because I had to check for the long cytokines to see whether
sorry, doctor, the last 4 minutes, please.
Pardon?
Last 4 minutes.
Oh, okay, the last 5 minutes, thank you. So the RSV, of course, I had to check for the long cytokines to see the IL-1, IL-4, IL-6, and then inflammatory cytokine to see actually whether this will actually be risen or not. Of course, and of course, the infection of RSV actually triggers immune system. So these cytokines will have to go up. So if you can see, IL-1 has gone up, even though there is no big significance between RSV and ampicillin good. The same goes for the IL-4 and IL-6. Even inflammatory FIN gamma, inflammatory gamma is also the same, has risen because there is a triggering for inflammatory cytokines. The same goes for ROT, which are T cell differentiation transcription factors. That is the B part. If you can see RORT, GATA, which is also one of those, FOXP, and then TBET, all increased. Okay, meaning there is an inflammatory response. Cytokines are going. Again, I had to go down to check for the microglia, meaning it's microglia phenotype, to see whether actually the RSV triggers something in the brain. So, remember, microglia is the brain immune cells. In the brain, one of the most common immune cells is microglia. They fight off anything foreign to the brain. So they actually triggers. We have to see that microglia are triggers. The phenotypic to determine whether this microglia will shift is IL-1 be better, and then INOS 1, and then IL-10, then arginine 1. So I had to do that too, and then realize that, yes, if you can look at it, they have increased, all of them, all of IL beta, INS, all increases like this, as opposed to the control and ampicillin. However, the messenger RNA for arginine 1 relatively shows a different trick, but which is very not much significant. So if you look at the image as well from the EBA INOS control and ampicillin, they are more or less the same. Okay, you can see the green is showing you the fluorescence, the green color here, and the red is actually showing the CD208 fluorescence. So this is also a significant thing. So it says that, yes, of course, the RSV virus can actually induce a shift of microglia in the microglia phenotype in the brain. So I had to check for the dysbiosis, the bacteriosis. I checked for the metagenomic sequence. So A, we could see that with the beta diversity among the group are normal. You can see normal control group is higher, no RSV, no problem. Lump microbiota is normal. But then with the normal control with the ampicillin, you see it has reduced. RSV also has reduced compared to RSV ampicillin. So it's like ampicillin RSV actually reducing the bacterial population in the pulmonary cavity. That is better diversity, right? Venn diagram is showing you that the differences of the diversity occur in different RSV groups infection, and of the results of the differential, you can see these are the heat map. You could show that infection cause disparities, and the tendency of this virus induced by RSV were among, and they are more obvious among ampicillin treatment. So ampicillin as antibiotics actually reducing the pulmonary cavity bacterial diversity, and the reduced diverse, they are the non-healthy they become. So we use the radar map actually to show whether these metabolites, what actually are their functions, what are their functions. The radar map shows that there are significant change. It shows that at least 10 metabolic differences among the groups were mainly function of cell membrane transport, carbohydrate metabolism, and also amino acids. So this actually is readable, and we did that. Study limitation, because it's a short-term study, it's a thesis, PhD, you don't need to have a very long time, there's no time. So we actually determined the sample size was too small, I believe. The time period in which we detect this is also less for long-term effects. So I call in for a more large sample size with a prolonged time to see actually the exact mechanisms. So but then, of course, when inoculating the RSV, it was done internationally. So probably there could be a neurological effect or inflammation through inoculation, during the inoculation, and that can also go through olfactory pathway. This is something that is a limitation. I would want follow-up studies to go on. While there's neurological infection is predicted to be responsible for the microbial phenotype change, a large sample size with a healthy neurological condition is recommended for these studies. In conclusion, this study was able to determine, or almost to find out that primarily microbes in the lung affect T1 or say T helper cell type immunoreactivity induced by RSV infection, and ultimately the lung microbiota induced microbial phenotype shift in the mouse brain or in the brain of the mice or tissue of the mice brain tissue. We were able to add scholarly information to the biological pathway called lung-brain axis through respiratory syncytial viral infection, the disruption of the primary microbiome, and the immune barriers that induce microbial phenotypic shift in the brain may be related mechanism in addition to the role of intrapulmonary bacteria on RSV infection. Moreover, the study focused on connecting the lung to the brain in the gut-brain axis biological pathway has associated effects of dysbiotic pulmonary microbes and some alterations in the brain. With these novel microbiome findings, their impact on human physiology, we recommend large sample size study with robust data analysis for the long-term effects of antibiotics and RSV infection on the brain tissues and physiology. With that, I'll tell you testicle and look forward to question and answers.
Very thankful, Mr. Dr. Osman. We are very happy for your presentation. Really, it was very good. I'm sorry because of the time. We have taken 2 more minutes. So for any question, if possible, we can have your email. You can send it personally in order to respect the time, if it's possible for you. Is it okay for you?
Yeah, it's okay for me.
Yeah. So we have to go to the next presentation, please. If there is any question, please, you can send it to the chat group. The Dr. Osman will be answered. And let's go to the next presentation, which will be presented by our brother, Dr. Shadman Tariq from Iraq.
Thank you very much.
Sharing.
And
Hello.
brother Shadman, we have 15 minutes for all the presentation, including the question, please. If you try to manage 10 minutes plus 5, it will be more better. Thank you.
Thank you very much. The presentation appeared?
Yes, it's appearing, please.
Yes. Thank you very much.
Welcome, please.
Dr.
The
Shadman,
project
welcome
was-
to our Congress.
Thank you very much. Hocam nasılsınız?
Sağ ol, sen nasılsın?
İyiyim Muhammed Şafi. Ne zaman geçti?
Hadi
Osman
kolay gelsin,
Bacınga.
görüşürüz gene.
Görüşürüz tamam. Osman Bacınga abi. Green biosensors of silver nanoparticle from wild mushroom Ganoderma lucidium, first time in Iraq. Nanotechnology has been well known as important field for research since the last century. Of the biological properties of nanoparticle from fungi is known as micro nanotechnology, which is usually made from fungal biomass and metabolites such as enzyme and proteins. In addition, one of the most important parts of the fungi cell is for manufacture of nanoparticle is cell wall. Generally, silver nanoparticle considered medically, medicinal, and environmentally safe, even if combined with other active group like the amino acid. Like other wide-distributed macrofungi in Iraq, the genus Ganoderma is considered one of the most important medicinally fungi due to the content of some species of active compound, about 400 active compound. The global famous species, Ganoderma lucidium, is known in Asia as the king of medicinal herb for its medicinal value due to the effect contained on the basis of the foregoing. The currently study aimed to prepare and characterize silver nanoparticle for the first time in Iraq from the large Iraqi wild mushroom Ganoderma lucidium material and method. This research, we applied many technologies and many devices, as well as some modified method. The mushroom was obtained from the college due to its wide distribution in Iraqi wild. It was available widely in our college and in our university. So the sample were collected from the university and the College of Science, characterized by my co-author, Dr. Sarah Kahtan Suleiman.
So, sorry, Dr. Shadman, I'm sure maybe you have some presentation, but what is appearing to me, it's only the first page. Is it okay for everyone? It's like that?
What?
Yes, that's right, Professor.
Only the first page is appearing. The other one is not appearing. If you want, maybe you're supposed to show it.
Is it not appeared?
Yeah, Shadman Bey sadece ilk sayfa görünüyor. Diğerleri görünmüyor yani. Şimdi görünüyor. Şimdi tamam.
Okay. Preparation of silver nanoparticles. Maybe in playing mode not appear in the screen share.
Now it's okay, I think. Preparation of silver nanoparticles.
Preparation of silver nanoparticles. Yeah. The first step was of preparation of silver nanoparticles was the aqueous preparation of aqueous extract from Ganoderma lucidium. Initially, the fruiting body of fungi were pre-dried, then grounding by electric grinder. Then 15 grams of grounded mushroom powder was dissolved in distilled water with vibrated bath, and their constant speed under constant speed and under consistent temperature for 48 hours. The obtained mixture was filtered firstly by Wattman and then additionally filtered by 0.22 micron filter to eliminate bacterial and other cells as a filtration, as a sterilization step. Preparation of silver nanoparticle. The aqueous that prepared of in the past slide, where the aqueous solution where of 1 millimole silver nitrate was prepared as the following: 0.169 gram of silver nitrate added to 100 ml of deionized water, heating the silver nitrate solution under 30 centigrade for half of an hour without continuous shaking. The binary were prepared by distilling 50 ml of mushroom extract prepared in the section 1 to 950 ml of 1 millimole silver nitrate. Purification of silver nanoparticle. The silver nanoparticle were purified, that contained silver nanoparticle prepared by in paragraph 2 was placed in Buffy tube, centrifuged in constant speed. The distillate was placed in centrifuge device again at the speed of 10,000 RPM for 10 minutes. This step was repeated for 3 times. The sediment is collected after drying and kept until used. Diagnosis of silver nanoparticle. The nanoparticle was diagnosed in 4 steps. The first step was in noticing color change, then spectrophotometer and FTIR, AFM, atomic force microscope, finally scanning electron microscope. Visual change. The optical and spectral characteristic of silver nanoparticles demonstrated by the observing the presence of color change when adding the aqueous extract of the fungi Goniodorma lucidum to the silver nitrate solution compared with the solution containing aqueous extract of the fungus without AgNO3. As you see, in the A is with the solution of nanoparticle with Ag, and the B without. The A part, the A tube, extract without AgNO3. The B is with Ag nanoparticle. UV spectra analysis. As you see in the slide, the highest wavelength was absorbance wavelength was of 450 nanometer, as shown in figure 2. This absorption peak was recorded also in several studies as Al-Salihi, Al-Faridi, and etc. The peak confirming the formation of the silver nanoparticle. When this peak appeared, it's confirmed that the silver nanoparticle is forming. Atomic force microscope. Atomic force microscope has been adopted as a useful tool for studying the morphological characteristics of biofunctional particle, as well as to scanning electron microscope. We applied atomic force microscope to study the functional group, biofunctional particle groups of the nanoparticle. The result of scanning electron microscope, as you see from this slide, that the particle ranged from 17 to 25 micrometer per particles. The conclusion: the projects demonstrate that eco-friendly and economically way for Ag nanoparticle biosynthesis using aqueous extraction of wild macrofungi, Gonoderma lucidium. As a result of this study, means that we can prepare a nanoparticle eco-friendly and economically due to its availability and wide availability in Iraqi wild. Obtained particle affected by several parameters like pH condition, concentration of material, reaction time, temperature, silver nanoparticle temperature. The silver nanoparticle produced in about 120 hours. The nanoparticle were highly stable. This is the important point when preparing any nanoparticle. The stability important due to save its activity in any medical or biological application. The result proposed potential utilization of a nanoparticle in medical application, especially in antimicrobial, anti-cancer treatment, and furthermore. I hope the idea is arrived to deliver it. Thank you for listening.
Thank you very much, Dr. Shadman Tariq. It was very, very important, and you have also respected the time. You have done 10 minutes for the presentation. Now we have 5 minutes for the question, please. If anyone want to ask any question about the green biosynthesis of silver nanoparticles from wild mushroom. If there is no one who want to ask, I want just to do some intervention to ask about the way you have done the drying. How did you dry the silver nanoparticle? It is my first question. Secondly, while doing the centrifugation of the nanoparticles, have you got some agglomeration status or no? Thank you.
Thank you very much. For the first part of question, the fruit body of macrofungi were firstly cleaned from dust and other particles, then dried under room temperature to not be effect on any active ingredient of fruiting body. Then grounded by electric grinder to obtain powder. Then this powder we can prepare any amount of nanoparticle solution by adding, as you see, 15 grams to 150 milliliter of distilled water. Then other steps that explained in this article. But for the second question, could you please repeat it?
Normally, while doing the centrifugation, when you did the centrifugation, did you find any agglomeration or no?
No, due to fine grounding of the fungi fruit body and complete dissolving, there was no agglutination or any macro during the centrifugation.
Okay, thank you.
Also, we as well as we filtered it in 0.22 micrometer filter. So the ground was completely and it was easy.
That's very good. I understood. Is anyone, there is 2 minutes more. If anyone want to ask any question, please. You can.
Dr. Shadman.
Efendim hocam, we will consume this 2 minutes to speak with Dr. Mohammed and with you.
You will speak with
Hocam,
Dr.
Nasılsınız? İyi misiniz?
Teşekkür ederim. Sen nasılsın?
Vallahi sizi çok özlüyoruz sadece.
Evet, Tikrit'te misiniz?
Evet, Tikrit Üniversite, Irak.
Tamam, çok güzel. Maşallah.
Vallahi hocam siz
Şimdi ben bir şey sormak istiyorum.
buyurun hocam.
Bu mantardan yapıyorsunuz. Başka ülkelerde de mantar oluyor. Onlardan üretmişler mi?
Var
Nanopartikül.
çok zaten Asya'da zaten çok var. Mehmet belki biliyor. Orada da çok var belki şey. Onlar diyorlar King of Herbs. O kadar değerli şeyler var, aktif ingredient var içinde. Kanser falan için kullanmışlar.
Özgür.
Ama Irak'ta da çok var.
Evet. Tamam, teşekkür ederim.
Teşekkür ederiz. Biz hocam yani bitki olarak nanopartikül üretiyoruz. Ama aynı şekilde mantar da yapılabiliyor şu an.
Evet.
Evet.
Tamam. I think the time is over. Thank you very much, Mr.
Thank you very much.
Dr. Shadman Tariq. Very happy to see you. Let's go to the next presentation. Our sister
Thank you.
Anisa Iktiarani, please, if you are here, start to share your presentation, please. Anisa.
Assalamualaikum warahmatullahi wabarakatuh. My name is Anissa Iktiarani. I'm medical student from Medical Faculty, University of Malang in Indonesia. And here I want to presentation about dihydrocarpene 1 from papaya leaf as an anti-inflammatory candidate that inhibits TNF-alpha receptor. This study aims to predict the potential of papaya leaf active compounds as an anti-inflammatory drug in in silico inhibition of TNF-alpha. The control drug used was hydroxychloroquine. Prediction of protein interaction used the ISSC application. The affinity of the active compound in papaya leaf to the TNF-alpha was predicted to use the molecular docking method. And hypothesis, active compound in carica papaya are predicted to be an anti-inflammatory candidate that inhibits TNF-alpha receptors. And then methods. First, read someone literature about anti-inflammatory candidate. And two, choose genus carica. And then three, search genus carica active compound in Dr. Group physical, chemical, and ethnobotanical database. And then search trigger and protein in PubChem, NCBI, and LSTB. And then docking in dockingserver.com and make some interpretation. The result from my first vitamin cholecalciferol, chlorogenic acid, dihydrocarpene 2, and beta tocopherol had free energy of less than -7 kilocalorie, but greater than control. Interaction surface are large except dihydrocarpene 2 and beta tocopherol. Of the four compounds, only chlorogenic acid has a high solubility. And then conclusion from the conclusion of the study is that the active compound of papaya leaf can be developed as an oral anti-inflammatory drug that inhibits the binding of TNF-alpha to this receptor. And then this research using the in silico method and to find the herbal potential of papaya leaf as an anti-inflammatory. That's all from my presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Sister Anisa, and thank you very much because the time also you have done almost 5 minutes with the presentation. Is there anyone who want to ask any question, please, regarding to our subject, please?
Anissa, may I ask you a question?
Yes, sir.
What is your department?
Is my department medical faculty.
Medical faculty.
Yes.
Which level? Undergraduate or master, PhD?
Yes, I'm a representation from Dr. Dini. Yes.
Okay, thank you.
Okay, Sister Anissa, I wanted to ask if possible, is there you can give some explanation about the TNF alpha receptors? Which kind of things? Why it should be inhibited, please? Thank you.
This research using in silico methods and to find the herbal potential of papaya leaf to as an anti-inflammatory, and then research on papaya leaf as an anti-inflammatory has not been TNF alpha, and then the active component and the papaya leaf as an anti-inflammatory that candidate that inhibit the TNF alpha receptor. Yes. Sorry, I'm sorry for my English not well. Now I'm learn to speak English.
No problem. Thank you. Thank you. There is no any problem. Thank you, Sister Anissa. Is there anyone who want to ask a question, please? Okay, I think the subject was very understandable. So we please try to go to the next one. Thank you very much, Anissa Ikhtiarani. And we have to go to our brother Yaizal Urganji, please. What's happened?
Yazat, mikrofonu da açarsan.
Tabii hocam, açıyorum hemen.
Merhabalar Yaizal.
Merhabalar.
Hoş
Teşekkür
geldiniz.
ederim. Hoş bulduk.
Buyurun kardeş.
Hocam burada bir saniye. Slide'ı alamıyorum şu an ama şu an şöyle yapayım o zaman. Sanırım izin gerekiyor. Ben şu an tam ekranı
Yok,
alamıyorum.
biz görüyoruz. Sen
Görüyoruz biz.
slideshow var yukarıda. Slideshow.
Sadece slideshow
File,
şey
Home,
yapsın.
Insert, Draw, Design, Animation, Slideshow. Üstteki menüde.
Hocam burada Mute, Stop Video, Participants, Chat, New Share var. Başka da bir şey yok.
Yağız. PowerPoint sayfasında menüye bak yukarıda. File, Home, Insert, Draw, Design. PowerPoint sayfasına bak. Biz görüyoruz PowerPoint'i.
Bir saniye hocam.
Sen PowerPoint'i görebiliyor musun?
Hocam şu an
Evet,
görebiliyor musun ekranımı?
Aşağıda var ya.
Geldi.
aşağıda-
Ekrana geldi. Sıkıntı yok.
Ha, şu an geldiyse sorun yok o zaman. Başlıyorum hocam. Hello.
Yağız, başlamadan Doktor Şafi, Züleyha Gadido gidip geliyor. Şu anda bağlandı. Eğer yapabilecekse onunla yapalım. Çünkü bağlantısı sürekli kesiliyor.
Okay. So
Züleyha,
Hocam hallettim şimdi.
devam edelim mi hocam mı? Yani bundan sonraki mi olsun?
tamam hadi bakalım. Bunu yapalım. Arkasından müsait olursa onunla yap.
Tamam
Okay.
hocam. Hello, I graduated from İzmir Cultural University in Mechanical Engineering. I'm currently working on a business analyst on the ETIA. I am doing on a master degree with Professor Dr. Mehmet Çevik.
Oh.
My master degree on business analysis and digital supply chain. Today I will present you Industry 4.0 based digital supply chains business analysis. I would like to say thank you about my teacher, Professor Dr. Mehmet Çevik, for the help for me. I hope I can create something in your mind about business analysis and digital supply chain. I am starting my presentation. I will talk about important points. The future of the manufacturing organization is being transformed globally by the development of the more digitized environment where value chains are interconnected and the production system because increasingly smart, autonomous, and autonomous. Industry 4.0 is a term used to refer to four industrial revolution characterized by the widespread use of the cyber-physical system in the modern manufacturing world. Digital disruption not only affects business models and the supply chains, it also significantly affects all segments of the society and including new relationship and the interaction with business and the people. While this study focused on the effects of the basic industry 4.0 technologies such as the Internet of Things, basic names IoT, cloud, cyber physical systems, reality, 3D printing, big data, and autonomous robots, we will relocate these studies to the business analysis methodology. That companies that want competitive in this condition must thus manage their supply chains and properly and efficiently. Real-time information exchange and the coordination among supply chain stakeholders is necessary for the efficient supply chain management. In the supply chain process, the digital supply chain makes the supply chain activities and enterprises of the easier and the more efficient. This photo shows us. In general, Industry 4.0 represents progress for the three important points. The one of the most important first point is the digitalization of the production, information system for the management and the production planning. And second one is automation, system using machines and the data collection from the production lines. Third of them, automation of the data exchange, other names the big data, linking production facilities in the comprehensive supply chain. Industry 4.0 relies on the adoption of the digital technologies to collect and analyze real-time data. It includes interoperability and the information transactionally, technical assistance, and the decreasing decision and the making. The other one is supply chain. Supply chain is structured of production where raw materials are converted into the final products and delivered to final customers. At the same time, the supplies have become much more complex, consisting of the large and complex network of the interconnected units. The final supply chain, I'm talking about that. Final supply chain includes all organization involved the upstream and the downstream networks of the product service finances and the information from the final supply to the customer. At the highest level of this customer, a supply chain consists of two basic interactive processes. The distribution of the logistic process determines how the product received and transformed from the warehouse to the retailers. These products can be transported directly and retailers initially and distribution facilities. This process includes the management of the inventory, taking, shipping, and the final product delivery. Supply chain management. Supply chain management is the actual management of the supply chain activities and examines customers' value and the achievement, sustainable competitive advantage. Organizations that make the supply chain are connected throughout the physical and the information flows and the form partnership that add value to customer experience. The greater emphasis and organization are the process flexibility, the need of the coordinate process and the many sites, competitive pressure and introduce new product faster. The digitalization process supported by information and the communication technologies has emerged from the driving force and business that have gained competitive advantage of the recent years. This is the Industry 4.0 missions in the future. I'm talking about the Industry 4.0 concept. The digital disruption not only affects the business model and the supply chains, it also significantly affects of all segments of the society, including new relationship and interaction with the business and the people. The Industry 4.0 relies the adoption of the digital technology collect to analyze real-time data and provide useful information to production system, the customer service in the entire value chain. And now I am talking about the IoT. IoT is the more popular time in the real world because of the IoT relies on the use of the internet access data from the remote sensor and the control physical objects of surrounding environment. IoT can be defined as the integration of the wireless sensor networks with the internet, having the efficient, the secure, scalable, and the market-driving compute to the storage research is the critical. Because of them, IoT is the framework. Now I am talking about cyber physical system. Physical systems are automation systems are the integrate innovation and the function throughout of the network. Industry 4.0 is based on the new production technology, new materials, the adoption, the diversified digital technology. Now, big data. Big data is required to create digital twins of the factory and then analyzes the provides ancient productive capacity and identifying the events that could impact production before they happen. Last, the important term is the cloud. Cloud is the production term. It can be represented by a combination of the various IT packages and including the internet service, web-based application, and the system administration. Defines the cloud bases the solution as the web-based application whose information is stored on external service over the internet. In the contrast, 3D printing relies the additive manufacturing, which creates the final products by creating the successive and layers of the material, thus eliminating the need of the assembly of the parts, this component. I'm talking about now robotics. In today's marketing, customers are looking the quick answers their request. Robots replace the hundreds of the human researchers and different organizations and provide cost advantage and business. Augmented reality. The Turkish name is the artırılmış sanal gerçeklik. It layers the digital content over the real life world, the Google Glass. This is the Google Glass. Virtual reality has the potential and dramatically change many aspects of the supply chain and the customer experiences and has been called the Internet of the experiences. This is talking about the IoT. Simulation. It can also be components assess the risk, cost, implementation, various impacts of the operational performance and roadmap to the Industry 4.0. This is the Meta. Facebook is new name is that Meta. System integration. System integration is about interconnected system components such as software and hardware. Other systems are subsystems. These components work together to provide the solution according to goals. Now, last one is the cybersecurity. Cybersecurity is the collection of the tools, police, security concepts, and risk management. Organization and the user assets include the connected computing device, personal information, telecommunication system, and the cyberspace. In Industry 4.0 and its effects, the reasons why Industry 4.0 medicine is benefits. It helps manufacturers and the current challenge by becoming the more flexible and reaction more easily change in the market. Industry 4.0 sustainable in the long run and helps to find solution and any challenges that arise. The conclusion. Industry 4.0 includes the use of many technologies such as the Internet of the Things, IoT, cyber physical system, big data, cloud, augmented reality, and big data. The implementation of the Industry 4.0 will provide many benefits and manufacturing experience. It is possible to list main benefits: Industry 4.0 flexibility, cost, efficiency, quality, and competitive and advantage. Thank you for listening my presentation.
Thank you very much, brother Isaac. It was very good, and you have tried also to respect the time. We have 3 minutes for any question, please. Three minutes, hocam. Okay, I think there is no question, please. Let's go to the next one, please. Hocam, shall we go to Zuleyha or shall we go by the list?
If Zuleyha is available, let's please continue with Zuleyha.
Okay, let's go with Dr. Zuleyha Gidado Mukhtar. Please, if you are here, you can start presenting your presentation, please. Thank you.
But I don't know if, Hojam, you can help me share the slides. I'm having challenges here sharing it.
Did you start your PowerPoint presentation on your computer?
I'm having challenges sharing it.
No, did you start PowerPoint?
PowerPoint. No.
Okay, then I will share.
Okay, thank you.
You will present it.
Okay, it's the last slide I shared.
Okay.
Thank you very much, Hojam.
Yes, you may continue.
Okay. Salam alaikum. Good afternoon, everybody. My apologies on behalf of myself and my colleagues for missing our time. It was a sincere misunderstanding of the time. I forgot, it was a miscalculation. So my name is Zuleyha Gidado Mukhtar, and on behalf of myself and my colleagues, I thank you all for the opportunity to present some aspects of our research titled The Optimization of Physicochemical Parameters in the Production of Yogurt from Tiger Nut and Bambara Nut Milk. So going to the introduction.
Your mic is off, Zuleyha, please.
Can you see Zuleyha?
Okay. So can you hear me now?
Yes, yes.
So should I start or should I go ahead from the introduction?
Yes, continue.
You continue with the introduction.
Okay. So as we all know, yogurt is a popular fermented dairy product consumed worldwide due to its inherent and well-known nutritional and health benefits. The high demand for dairy products, particularly yogurt, has increased significantly over the years due to these perceived health and nutritional benefits. However, in recent times, there have been an increased demand for non-dairy yogurt due to dietary restrictions, allergic concerns, and personal preferences. So these are pictures showing some of the type of yogurts we have made from products we have made from yogurt. You can see that they are varied and wide. So based on these concerns regarding the high cost, limited availability of cow milk, that is the dairy milk, and then the health concerns, it has led to more interest or search for alternative sources of non-dairy milk to produce yogurt. So from where I am, we have tiger nuts and bambara nuts, which are endemic wild fruits, which we gather from the wild. So they're more or less free, they're cheap, they're available, and they're almost available throughout the year. So because of that, we looked at them to produce milk, and as they have also been reported to have some nutritional and health benefits, to seek ways to explore their potential as an alternative source of cow milk in yogurt production. So this, the first picture is the picture of tiger nuts for those who are not familiar with it. And this, the one on the right is bambara nut, which are both endemic to my locality here in Nigeria. So next slide, please. So more or less the aim of our research, which is a preliminary research, which we hope to be a more wider scoped project, is to produce yogurt from tiger nut and bambara nut milk and to optimize the production process of producing yogurt using these non-dairy sources of milk. So the objectives to determine the ideal concentration of tiger nut and bambara nut milk for yogurt production, and to determine the ideal pasteurization temperature for yogurt production from these non-dairy sources, and to evaluate the quality and stability of each formulation to establish the ideal formulation and fermentation with best sensory qualities. So this is the method we use. It's a schematic diagram so that everybody can see the step-by-step process we followed. So we got our nuts, we sorted them, we soaked them, they were drained, washed, and cleaned carefully. Then they were milled to get milk from tiger nut and bambara nut. So the next slide, please. So this is the experimental design. B represents the bambara nut, while T is for the tiger nut milk. So we use different concentrations and different formulations where we either had the bambara nut milk on its own or the tiger nut milk on its own. Then we had a mixture of them in different ratios to see what we could come up. We also use different concentrations, weight over volume, pasteurization temperature. You can see the temperatures we use, and then different pasteurization times and different fermentation temperatures and different fermentation times. So based on our results, we have the physicochemical parameter we are presenting here is the pH. So you can see from B1, T1, BT1, B2, T2, and BT2, we can see that the pH range from 6.3 to 6.8. Then we assessed their sensory qualities, and we found some had good acceptability, some were not acceptable as the others. Please, can we have the next slide? So based on the pH result and the sensory evaluation, the yogurt we were able to produce from these non-dairy sources were found to be generally acceptable. The pH values of all the formulations were within the acceptable pH range for standard yogurt, and so also the sensory evaluations revealed good acceptability for the B1 formulation, BT1 formulation, and B22 formulation, while there was good acceptability for B2, likely acceptability for T2, and non-acceptability for T1. So in comparison to a study by Adengoa et al. In 2021, who evaluated the physicochemical and sensory properties of yogurts from also tiger nut and cow milk, our study produced similar results in terms of acceptable pH range and sensory evaluations for the tiger nut-based formulations. So next slide, please. Another study by Oluwale et al. In 2018 investigated the physicochemical and microbiological properties of yogurts produced from blends of bambara nut and cow milk. Our study produced similar results in terms of the acceptability of the pH and sensory evaluation for the bambara nut-based formulation. However, their study did not evaluate formulations using only tiger nut milk or combination of both the tiger nut and the bambara nut milk. So in conclusion, the study results showed that all formulations produce curdled yogurt with acceptable pH values and good sensory acceptability. And then we can conclude that bambara nut and tiger nut milk produce the most preferred yogurts based on the sensory evaluation we conducted. Then the findings also suggest that the combination of bambara nut and tiger nut milk can enhance the sensory properties of yogurt, as we know that is the dairy dairy yogurt. So next slide, please. So we can recommend that based on the results obtained, that bambara nut and tiger nut milk can be used as alternative sources of milk in yogurt production, and the combination of the two nuts in a 40% concentration and fermented at 42 degrees for 7 hours produce the most acceptable yogurt in terms of sensory evaluation. Further studies could also focus on the health and nutritional benefits of the combination of these nut-based yogurts, and efforts could be made to promote the consumption of this yogurt as a means of supporting local agriculture and also increasing the potential of our wild foods, which are more or less not used for anything, and they are becoming more extinct by the day. And it could also be used to improve food security and nutritional security, which is, as we all know, a huge problem for us in Africa, especially sub-Saharan Africa. So thank you very much for listening.
Very thankful, Dr. Zuleyha. It was very, very, very understandable. You have understood the subject, and you have also respected the time. You have almost 9 minutes for the presentation. We have 6 minutes for any question, please. Thank you.
Can I ask a question to Dr. Zuleyha?
Yes, please,
Yes.
Doctor. Professor, please.
In fact, it's very interesting.
Yeah.
I want to ask about the cost, the price, because milk is not very expensive in Turkey, but not milks, as I heard, are very expensive in Turkey. What about in Nigeria?
No, in Nigeria, like this tiger nut and bambara nut. In fact, bambara nut, all you do is when it is in season, they just go into the wild areas because we have land mass where the trees are. They are not even farmed in the form of agriculture. They just grow wild in the forest area. So they just gather them. So they are more or less inexpensive. That is why we are considering an alternative source, because cow milk here is very, very expensive. If, for example, cow milk a liter, you can get it for maybe $5. This you can get it for about $1 or less.
Strange. You say in Turkish we say, or in Arabic, Hüdayin abi. Do you understand it?
No, I don't.
Hüdayin abi, that means it's a gift of God.
Definitely, alhamdulillah.
Yes.
Myself,
presentation.
Thank you very much. Also, as I said, it's very interesting subject. I have one more question, please, Dr. Zuleyha. What about the pasteurization temperature? I think you have mentioned 90 degree. Is it, why 90 degree, and is it not harmful to the contents of the milk?
Well, we tried different temperatures, and based on the study,
I'm sorry, I think the network has gone.
Okay.
I think the network has gone from Zuleyha, but I think my question was answered. There is no problem. Thank you very much. Once again, Dr. Zuleyha Gidado Mukhtar. Let's go to the next one. And I'm sorry for Muhammad Ali and Musa Ali
that
Hassan
have been
Kroma,
conducted
because
before.
we have jumped, we have changed, we have taken Zuleyha because she has some network problem. I think you can understand what we have done, and we are apologizing. Let's start with our brother Muhammad Ali. Please, if you are here, we are listening to you. Thank you, Zuleyha. I have understood the question, please, the answer.
Okay, thank you.
Welcome, please. Muhammad Ali.
If he is not here, then let's continue with Kroma, and someone else has entered, just entered, Butsenia, and the final presentation will be from Butsenia. Let's continue with Kroma.
Okay, let's let's continue with our brother Musa Ali Hassan Kroma. Please, I think you are here. We are listening to you, please, bro. If your mic is maybe closed, maybe because we don't hear you.
It's open.
Okay. Welcome, please, Musa, bro.
Thank you so much. I hope you are getting me. Am I audible?
Yes, everything is okay.
Okay,
the presentation is clear and the voice also is clear, please.
Good. Hello, everyone. My name is Musa Al Hassan Kroma. I'm a student at Chukwuemeka University in Adana, where I study biology. I am glad to be here today to present my research. So before I proceed, I want to let everyone know that this is part of my undergraduate project that I'm doing in my university for graduation. And I'm glad to be presenting to you guys.
Very good.
So my presentation is divided into sections, which include the introduction, the objective, the methods, results, and the conclusion with references. So these are the keywords. A minute. Good. So as you all saw from the beginning, my research is based on the evolutionary biology of infectious diseases. And a year ago, we had a pandemic, which caused every one of us to go into lockdown. It affected our schools, affected businesses, it affected transportation. In general, it affected our life. So I'm just trying to say that infectious diseases have shaped our life, or have shaped things that go on Earth into different ways. It evolves in so many ways, and it comes in different shapes. So understanding their evolution, understanding the mechanisms that cause this evolution into those diseases is very important. And it will help us to manage future outbreaks, and also to know on how to respond to them if we get an outbreak or pandemic or epidemic in the future. So the objective of this research, firstly, I want to also let you know that the research is totally literature-based. That's the first stage of it, because I want to expand upon it in the near future, maybe for my master's. And for that, I plan to use a software to run a simulation of the activities using the evolutionary principles to analyze this evolutionary biology of infectious disease. But for now, it's totally literature-based, which is a secondary research. So it aims to explore the evolutionary biology of infectious diseases and to understand the mechanisms that drive the emergence and the spread of the diseases. So while conducting the research, the method we use, well, we conducted a comprehensive literature review on current and past papers on the topic. And we do that by using PubMed, Web of Science, online journals, and database to identify relevant articles, research papers, and other necessary sources. We use keywords such as infectious disease, evolution of infectious disease, biology of infectious disease, infectious diseases and evolutionary forces, molecular biology of infectious diseases. We use these keywords to be able to gather every or at least available papers or articles on the topic. And then we examine those papers and those articles, and we got the rightful information to use for this research. So the result we got upon doing that was that we understood that infectious disease involved in response to selective pressure imposed by host immune system or population demography. According to the Handel and Reinberg evolutionary equilibrium, it says that a disease or a genetic will remain constant, it will remain equilibrium constant until there's a force acted upon it. For example, COVID-19 came, it came with one strain, but because of other forces, other evolutionary forces or environmental forces, it started to evolve into different strains. Then we had the Omega and the other different types start to come up. So this was clear to all in our research. And the second result we got was pathogens get involved in a variety of strategies to invade host immunity, such as antigenic variation and immune subversion. And then we also got another result, which is genetic variation plays a crucial role in the emergence of new pathogens, like I said earlier. And also horizontal gene transfer contribute to the evolution of antibiotic resistance. So I'm going to give you some two or three case studies. For example, we have the influenza virus, which is a highly mutable virus that evolved rapidly to invade host immunity or the immune system. Is antigenic drift and antigenic shift are one of the mechanisms of immune evasion. And it also has genetic reassessment, which contribute to the emergence of new strains. Another example is the Plasmodium, which is a cause-active agent of malaria, and it's evolved into to multiply drugs. Evidence suggests that horizontal gene transfer has or can contribute to the evolution of this of its resistance to drugs. And then the last case study for the research is HIV. We all know that is a retrovirus that affects the human or animal immune system. It evolved rapidly and has high genetic diversity, and it mutates. Mutations and recombinations contribute to the emergence of drug resistance when it comes to HIV. So in conclusion, the emergence and spread of infectious diseases are driven by complex interplay of genetic, demographic, and ecological factors. And also understanding the evolutionary biology of infectious diseases is crucial for developing effective disease control strategies, especially for developing countries like Liberia or countries in Africa. And this research will be very helpful to those countries. Thank you so much.
We are thanking you very, very, very thankful, brother Musa Al Hassan. And also you have tried also to respect the time. You have done almost 7 minutes into the presentation, and we have 7 minutes for any question, please, dear auditor. Hocam, var mı bir sorunuz?
Teşekkür ederim Hasan Kroma. Sanıyorum başka
Rica ederim.
katılımcı kalmadı. Birisi vardı ama o bir türlü iletişim kuramıyor.
Kroma yüksek lisans yapıyor musunuz şimdi?
Hayır, lisans okuyor.
Ne zaman devam edecek misiniz yoksa gideceksiniz?
Yani devam etmeyi düşünüyorum da.
Tamam. İzmir'e gel bence.
İnşallah yaparım.
İnşallah. Tamam. If there is no question, please. Let's go to Sister Hussein.
Together.
Huh?
Let's take a screenshot all together.
Buseina is not going to present, hocam?
She dropped. She's not here.
Okay.
She or he, I don't know.
Buseina, she. That means Buseina. She's female.
Buseina, but name is Saad. Saad Buseina. Saadi, I don't know.
Saad o
Musa,
mail olabilir.
Senin paylaşımı kapatabilirsin.
Tamam hocam.
Arkadaşlar, müsait olan herkes kamerasını açabilir mi? Anissa, please start your camera. Osman,
Züleyha, your camera. Dr. Osman, if you are here, please.
Kavsu. Is there anyone?
Hello, I'm here.
Okay. Kavsu, hoş geldin. Hasan Kroma
let's go
is
soon.
double, cloned.
I was afraid that my computer will go off. So I entered double.
If everybody is ready, I'm taking. That's okay.
Thank you. Thank you very much.
Thank you.
And was very happy to be in this session.
Thank you very much.
Helpful. Thank you very much.
Thank you very much.
Dear participants, as the chair of the Congress, I want to thank all of you.
Thank you very much.
It was a
hocam.
nice program of 2 days, and this is the last final session, and we are ending the Congress today.
Very good.
Hope to see you, inshallah, in our next Congress next year.
Thank you. Thank you very much, Mohammed.
Thank you. Selam.
Thank you.
Aleyküm selam.
Thank you to all the participants. Thank you.
Thank you very much.